Dear students, today we are going to discuss. Dear students, today we are going to discussing about genetic code. Genetic code is a dictionary that corresponds with sequence of nucleotides and sequence of amino acids. Words in dictionary are in the form of codons. Each codon is a triplet of nucleotides. 64 codons in total, and 3 out of these are nonsense codons. 61 codons for 20 amino acids. Introduction and genetic code. The letters A, G, T and C corresponds to the nucleotides found in DNA. They are organized into codons. The collection of codons is called genetic code. For 20 amino acids, there should be 20 codons. Each codon should have 3 nucleotides to impart specificity to each of the amino acid for a specific codon. Nucleotide 4 combinations, nucleotide 16 combinations, nucleotide 64 combinations. Most suited for 20 amino acids. This is the genetic code. Pre-bases code for 1 amino acids. Initiator codon. AUG is the initiator codon in majority of proteins. In a few cases, AUG may be the initiator codon. Methionine is the only amino acid specified by just one codon, AUG. Clinical significance. Clinical significance, mutations can be well explained using the genetic code. Point mutations, silent and missense, nonsense, frame shift mutations. Oblink phenomenon. First, 2 bases in codon in mRNA 5' to 3', base pair traditionally with the 2 and the third base of the anticodon in tRNA 5' to 3'. Non-traditional base pairing is observed between the third base of the codon and the first base of anticodon. The reduced specificity between the third base of the codon and the complementary nucleotide in anticodon is responsible for oblink. Genetic code universal. Universal in all living organisms, genetic code is the same. The exceptions to universality is found in mitochondrial codons, where AUA codes for methionine and UGA for tryptophan instead of isoleucine, and a termination codon respectively of cytoplasmic protein synthesizing machinery. AGA and AGG code for arginine in cytoplasm, but in mitochondria they are termination codons. Genetic code characteristics. Specificity. Genetic code is specific. A specific codon always codes for the same amino acid. Examples: UUU codes for phenylalanine. It cannot code for any other amino acid. Oblink phenomenon. Proline has 4 codons: CCU, CCC, CCG, CCA and CCG. The first 3 codons can be recognized by a single tRNA having inosine at the first place, IgG and 5' to 3'. Genetic code, non-overlapping and non-punctuated. All codons are independent sets of 3 bases. There is no overlapping. Codon is read from fixed starting and point as continuous sequence of bases taken 3 at a time. The starting points is extremely important, and this is called reading frame. Genetic code redundant. Genetic code is redundant, also called degenerate. Although each codon corresponds to a single amino acid, but a single amino acid can have multiple codons. Except tryptophan and methionine, each amino acid has multiple codons. UCAG ఇక్కడ కనుక మనం అబ్జర్వ్ చేస్తే UUU, UUC, UUA, UUC, UUA and UUG. First 2 codons phenylalanine and leucine. Next one is UCU and UCC, UCA and UCG. These are amino acids. This codons codes for amino acids. This is the table of genetic code. First letter and this is the third letters. Genetic code summary. Genetic code is unambiguous, universal, degenerate, complex, and non-overlapping. UAG, UGA, and UA are termination codons. AUG is the start codon. Mutations can be well described using genetic code. Nonsense codons. There are 3 codons out of 64 in genetic code which do not encode for any amino acid. These are called termination codons or stop codons or nonsense codons. The stop codons are UAA, UAG, and UGA. They encode no amino acid. The ribosome passes and falls of the mRNA. This is the second letter, this is the first letter, this is the third letter. Thank you.